Okay, so this is ATP. You're probably very familiar with ATP right now. The important thing to know here is that ATP is adenine triphosphate, and the phosphate groups that contain a lot of energy in those bonds. So when that phosphate-phosphate bond is broken, we get energy that is released. When that bond is broken, it's going to release that phosphate group and it will become ADP. Think of ADP as a battery that has been drained and needs to be recharged by becoming ATP in order to be used for energy again in the cell. There are three different mechanisms that bodies use or cells use to create this vital ATP, which is the energy that the cell must run on. The first mechanism is substrate level phosphorylation. This occurs when a phosphate group is transferred from some other molecule and it gets put on ADP to create ATP. The second method is oxidative phosphorylation. This is when the phosphate is added to ADP, but this is done through a series of oxidation reduction reactions that occur during a cellular respiratory pathway. We see this, for example, in the electron transport chain, which is the last step in the cellular respiration process inside the cell. We also have a method of, not in our bodies, but in a lot of autotrophs, we have photophosphorylation. This is when phosphate groups are able to be added to ADP in order to make ATP using energy collected from sunlight. There are three metabolic strategies that we see cells use in order to get the energy that they need to make ATP. The first is aerobic cellular respiration, which in a nutshell is glycolysis, followed by the citric acid cycle, and finally ending up with the electron transport chain. With aerobic cellular respiration, this requires oxygen, and oxygen is actually used as the final electron acceptor in the process. This is a little bit different from anaerobic cellular respiration. However, anaerobic cellular respiration still goes through the same glycolysis process, the same citric acid cycle, and pretty much the same electron transport chain cycle as well. The difference, though, is that instead of using oxygen as the final electron acceptor, another inorganic molecule besides oxygen will be used as the final electron acceptor. Some examples of this are iron compounds, sulfates, nitrates, and nitrites. The third is fermentation. In the fermentation process, this includes undergoing glycolysis only. It does not undergo the citric acid cycle or the electron transport chain. An organic molecule is used as the final electron acceptor versus other strategies that always use the an inorganic electron acceptor. For example, when our muscles do this, we make lactic acid and we actually use pyruvate as the final electron acceptor in our muscles when they undergo lactic acid fermentation. Oxygen is not needed for anaerobic cellular respiration or for fermentation. I'm just going to have to edit that out, but hurry, because I don't want to screw it up. Okay. All right. There are three metabolic strategies. So the aerobic respiration is the most efficient. It can produce up to 38 ATP. We've got anaerobic respiration, which depending on the methods used, it can vary between producing 2 ATP all the way up to 36 ATP. The fermentation process is the least efficient of the metabolic strategies and produces a maximum of only 2 ATP. Here's a couple of categories that you need to know that are based on how energy is obtained by the prokaryote or the organism in general. So we've got autotrophs versus heterotrophs. So organisms that can synthesize their own food from inorganic sources are called autotrophs. For example, we see that some bacteria cells or prokaryotes can actually use carbon dioxide in the air as their carbon source. Heterotrophs, on the other hand, are organisms that cannot synthesize their own food. So these organisms must consume organic molecules in the forms of sugars, proteins, or lipids. Here is a summary of organism classifications based on energy or electron sources. So let's look at some of these to make sure that we understand. Chemotrophs are going to either be chemoheterotrophs or chemoautotrophs. Let's first look at chemoheterotrophs. Chemoheterotrophs are also called chemoorganotrophs. These organisms are going to use organic chemical compounds as their electron source. These are proteins, lipids, and carbohydrates. The chemoheterotrophs that are able to use oxygen are going to undergo aerobic cellular respiration. The chemoheterotrophs that are not able to use oxygen will undergo the process of anaerobic cellular respiration. Chemoautotrophs, or chemolithotrophs, have the ability to use carbon dioxide as their source of carbon. They use inorganic chemical compounds such as the electron acceptor sulfur, iron, carbon monoxide, and hydrogen as their final electron acceptors. Phototrophs have the unique ability to use sunlight as their energy source. Phototrophs that use organic compounds like proteins, lipids, and carbohydrates as a carbon source are called photoheterotrophs. Phototrophs that use carbon dioxide as a carbon source are called photoautotrophs. Photoautotrophs that release oxygen as a waste product are called oxygenic photosynthesizers. And photoautotrophs that do not release oxygen as a waste product would be called anoxygenic photosynthesizers. Here are some interesting energy sources that some of these chemoheterotrophs or chemolithotrophs can use. So chemolithotrophs are able to use inorganic chemical compounds as their electron source. We see some of these inorganic compounds are extremely toxic to other life forms. We see, for example, toluene, which is used as an industrial solvent, ammonia, which is used as a cleaner, and arsenic, which we know is a poison for humans. This is just a summary of the flow chart that we saw earlier, and I wanted to just add that in case you wanted to print that out or use it as an additional study guide. Okay, so let's take an overview of cellular respiration. So in cellular respiration, it has three processes, but it also has one vital transition reaction that in microbiology, especially since that transition reaction produces something that we're interested in, which is NADH, we like to add that here, even though it's not really considered a separate, well, it's not really considered a process. If you look at almost any textbook, a lot of times will just show three processes that are involved here and omit the transition reaction. So, but I will be talking about that as well for completeness sake. So here we see the overall reaction of cellular respiration. In a nutshell, we see that glucose and oxygen are going to be used in order to make carbon dioxide, water, and ATP. Glycolysis is performed in all living organisms. Glycolysis always takes place in the cytoplasm of the cell. Glycolysis does not require oxygen and can be performed in both aerobic and anaerobic prokaryotes. Glycolysis can be broken down into two phases: an energy investment phase, in which 2 ATP molecules are used to start the glycolysis process. Later in glycolysis, we get an energy payoff phase. In the energy payoff phase, 4 ATP molecules are produced. The net gain for glycolysis then is 2 ATP molecules. Glycolysis begins with 1 glucose molecule. Glycolysis produces 2 ATP net. Glycolysis also produces NADH, which performs the vital function of carrying electrons to the electron transport chain. The end product of glycolysis are 2 pyruvate molecules. There are 3 glycolysis pathways. The glycolytic pathway used by most organisms is the Embden-Meyerhoff-Parnas pathway, or EMP pathway, which is what we were just looking at. But there are also the ED pathway and the PPP pathway. The ED glycolysis pathway is interestingly the only form of glycolysis used by the gram-negative pathogen Pseudomonas aeruginosa. Pseudomonas aeruginosa is a common bacteria known for being antibiotic resistant. This pathogen causes many of the hospital-acquired infections known today. Also, E. coli is able to use this pathway, but it can switch as needed to the EMP pathway as well. The last pathway is very important for all organisms. This is the pentose phosphate pathway, or PPP glycolysis pathway. The PPP glycolysis pathway creates building blocks or precursors that are used for amino acids and nucleotides. This form of glycolysis will take place anytime the cell is in need of amino acids or nucleotides. Before a pyruvate molecule can enter the Krebs cycle, it first may undergo a transition reaction. After glycolysis, each of the pyruvate molecules that were produced will be transformed into acetyl-CoA. During this process, we have carbon dioxide released and NADH is produced, but no ATP is produced in this step. After the transition step, acetyl-CoA will enter the Krebs cycle. Here in the Krebs cycle, we will have more NADH and FADH2 produced. 1 ATP is also produced by substrate level phosphorylation. Also, we are going to get more carbon dioxide released. So keep in mind that 1 glucose molecule will take 2 turns or 2 cycles of the Krebs cycle in order to be fully processed. The last process in cellular respiration is the electron transport chain, sometimes called oxidative phosphorylation. The ATP formed in the electron transport chain, or ETC, is formed using oxidative phosphorylation. There is an enzyme that is needed for this process, and this is the ATP synthase. The ATP synthase is in essence what creates the ATP. The electron transport chain exists in the plasma membrane of prokaryotic cells. Let's look briefly at the steps of the electron transport chain process. So all of the NADH and FADH2 that were generated in the previous steps of cellular respiration. This includes glycolysis, the transition reaction, and the Krebs cycle or citric acid cycle. So the NADH and FADH that were created from these different processes will now deliver those electrons that they gained from those processes. They're going to deliver the electrons to the electron transport chain. Once at the electron transport chain, let's look at what happens. The hydrogens are taken off of the NADH and the FADH2, and they will go to the first complex of the electron transport chain. Now, I need to give you a little bit of chemistry here so you understand what's going on. Hydrogen is made up of one proton and one electron, which hopefully you remember. The electron is going to get pulled off here in the electron transport chain. So now the proton and the electron are going to be separated from one another. It is the electron is going to go through the different complexes in the electron transport chain. Now, what is left over is the hydrogen ion. Once hydrogen loses its electron, it becomes H+ or hydrogen ion. In essence, this is just a proton. Protons are pumped out of the membrane using a proton pump. So they're going to be pumped out of the membrane and they're going to go into the space between the membrane and the cell wall. So the proton pump and the action of this proton pump is going to create what's called a concentration, or more specifically, an electrochemical gradient. The proton pump will move protons from inside the cell to outside the cell, between the cell wall and the cell membrane. The protons will want to get back into the cell wall due to its electrochemical gradient. This is called chemiosmosis. This forces the protons to travel through the ATP synthase in order to get back to the cell. As the protons, or H+, travel through the ATP synthase, this is going to give off the energy that's needed for the ATP synthase to make ATP. In aerobic respiration, oxygen is going to be the final electron acceptor in the terminal step of cellular respiration. A single oxygen molecule is going to catch two electrons that are coming off of the end of the electron transport chain. The oxygen molecule is also going to pick up two protons from the surrounding intracellular fluid. This means oxygen is adding two hydrogen molecules to become water. In anaerobic respiration, a different inorganic molecule, such as nitrates or nitrites typically, will be used as the final electron acceptor in the terminal step of the electron transport chain. Obligate anaerobes will use anaerobic respiration. The cell also has processes that it uses to break down or catabolize proteins and lipids. The important thing to know here is that these other pathways will merge with the pathway of cellular respiration, as shown here. But what about fermentation? So let's just revisit this again. So fermentation is not nearly as efficient as aerobic cellular respiration or anaerobic cellular respiration, as shown here. And fermentation only gives us about 2 ATP per glucose molecule consumed. Obligate anaerobes are prokaryotes that require oxygen for their metabolism. Humans are an example of obligate aerobes, since we absolutely depend on the presence of oxygen. Obligate anaerobes are prokaryotes that do not need or cannot be exposed to oxygen, and these only undergo anaerobic metabolism, or anaerobic cellular respiration. These are classified as obligate anaerobes. Botulinum toxin is one of these. Interestingly, we also get facultative anaerobes. These prokaryotes have metabolic flexibility. They have metabolic pathways that they use when oxygen is present, and a different set of metabolic pathways it can use when oxygen is gone or absent. An example of a bacteria that uses this is the bacteria that is responsible for staph infections and strep throat. Alcoholic fermentation is one of the types of fermentation. So these organisms do not require oxygen. Fermentation is going to take place in the absence of oxygen, and it's going to use an organic molecule such as pyruvate as a final electron acceptor. Remember, these only undergo glycolysis. Fermenting organisms produce 2 ATPs per single glucose molecule. The yeast Saccharomyces cerevisiae is the microbe that is commonly used for alcoholic fermentation, baking, or even biofuel production. Now, this is not a bacteria, this is actually a yeast. What is a yeast? Yeast are single-celled fungi that belong to the fungi kingdom in the eukaryota domain or eukarya domain. This means they're eukaryotic. A different type of fermentation is lactic acid fermentation. Fermentation by some bacteria, like those used in yogurt and other soured foods, and by animals in mussels during oxygen depletion, like our own mussels do, is the process called lactic acid fermentation. Lactic acid bacteria are a special type of bacteria that are known to use lactic acid fermentation. We can just call these LAB. Many LAB are gram-negative, and they're used in food production for some types of yogurts and cheeses. A different type of organism is actually able to undergo heterolactic fermentation. Heterolactic fermentation means that these cells are able to produce more than one fermentation product. We actually use these types of bacteria to undergo heterolactic fermentation to make pickled foods like our pickle. The fermentation process gives off carbon dioxide gas as a waste product. This means the bubbles in your champagne or beer and the holes in your bread and cakes are all due to little tiny bubbles made by carbon dioxide that was released as a waste product in the fermentation process. There are a few enzymes that you should know, and they have convenient names. We have lipases that function to take apart triglycerides. They're going to take apart triglycerides into their glycerol component and their fatty acid subunits. Phospholipases are able to disassemble phospholipids, breaking apart the phosphorylated head unit from the individual fatty acid tails. Beta oxidation is a process that disassembles fatty acids. Proteases disassemble proteins by breaking the larger proteins into smaller amino acid segments that are called peptides. Lastly, one word about photosynthesis. Have you ever noticed that the chemical equation for aerobic cellular respiration and photosynthesis are basically the same but in reverse? So we see here that the reactants of cellular respiration are the products of photosynthesis, and the products of cellular respiration are the reactants in photosynthesis.